friends, hope all are doing well. Welcome to my YouTube channel, Sam Biology Class. Friends, today we will discuss about PIP2 mediated intracellular signaling pathway. But before that, please consider subscribing my YouTube channel, and when watching, don't hesitate to share your comments. If this video is useful, then like and share it with your friends. Eukaryotic cells contain three main categories of surface receptors. Number 1, G protein coupled receptor or GPCR. Number 2, ion channel linked receptor. And number 3, enzyme linked receptor. All these receptors are integral membrane proteins that remain embedded in the cell membrane. These receptors bind with specific extracellular ligands and convert it into internal signals. PIP2 mediated intracellular signaling pathway, also known as PIP2 mediated intracellular signal transduction, involves specific extracellular ligand, G protein coupled receptor, heterotrimeric G protein, a cell membrane enzyme known as phospholipase C, and a minor cell membrane phospholipid component, that is phosphatidylinositol bisphosphate or PIP2. PIP2 is the substrate for a number of important signaling proteins. It also forms lipid clusters that sort proteins. This is the heterotrimeric G protein belonging to GQ family. This protein consists of three different subunits such as alpha, beta and gamma. In inactive state, alpha subunit remains bound to GDP and all these three subunits remain together and are associated with the GPCR. Like other surface receptors, GPCR also binds with specific extracellular ligand such as hormone, neurotransmitters and others. This binding induces conformational changes in the receptor, which then act as a nucleotide exchanging factor. The activated GPCR exchanges the GDP, which remains bound to the alpha subunit, with GDP. Binding of GDP activates the alpha subunit, which is then dissociated from beta-gamma dimer. The activated alpha subunit then stimulates a cell membrane enzyme known as phospholipase C. Phospholipase C then cleaves phosphatidylinositol bisphosphate into diacylglycerol and inositol triphosphate. Both act as second messengers. Diacylglycerol remains associated with the cell membrane and activates an enzyme known as protein kinase C. Like other protein kinases, this enzyme also phosphorylates different proteins that leads to response in the cell. Protein kinase C is an important enzyme that is involved in receptor desensitization, in modulating cell membrane structure events, in regulating transcription, in mediating immune response, and in learning and memory. The second cleavage product is inositol triphosphate, which diffuses into the cytoplasm and then activates calcium ion channel, which are present in the membrane of endoplasmic reticulum. This activation results in opening of the calcium channel, as a result of which calcium ions are flooded from lumen of ER into the cytoplasm. This raises calcium ion level in the cytosol. The calcium ions bind with calmodulin. Calmodulin is a calcium binding protein. Binding of calcium ions induces conformational change in the calmodulin, which then activates different types of protein kinase, which in turn phosphorylate specific proteins that regulate contraction, metabolism, and transport. So friends, this is all about PIP2 mediated intracellular signaling pathway. For more updates, subscribe my YouTube channel, Sam Biology Class.